Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture I am going to talk about T cell activation and differentiation. In the previous videos lecture, if you guys remember, we talked about T cell biology overview and we talked about T cell receptor, types of T cell. In types of T cell we talked about helper T cell, cytotoxic T cell, and we also talked about T regulator cell, memory T cell. All these things we talked about. Okay. Now this is another video lecture which is actually regarded to the T cell biology, and in this video lecture we are going to talk about T cell activation, that how it activated and how once it activated and how it will be differentiated into other cell. We'll be talking all in this video, so please stay tuned and keep watching. Let's start. Okay friends, so now let me write. Okay friends, first of all let me talk about a brief introduction. Let me first of all explain a brief introduction about the T cell activation and their differentiation. Then we will explain it through diagram. What actually happen in disease condition when the microorganisms internalize inside a body through ingestion or injection, any kind of thing. If the microorganism get internalized inside our body, then macrophages will engulf it, or the normal cell become infected, let's suppose. After that way, infection will be occur, and after the infection, disease condition will be happen. So after that way, if we want to control all this infection and disease, so what happen inside our body, the T cell will be activated. So if we want to activate immunity against this infection, so the T cell will be activated. Activation of the T cell is very important for the control of each infection and disease. Okay, so here the question arises in our mind that how it will be activated. T cell will be activated by three pathway. For the first time, that what happened, first of all, the antigen presenting cell will engulf the pathogen or bacterial cell or any microbes. They first engulf that. Once they engulf that, so then they will convert into small fragment. After that, then they will represent this small fragment to their receptor, which we call MHC2 receptor or molecule. After that, they will produce cytokines due to which the T cell will be attracted, a naive T cell. Once the naive T cell is attracted, so then this naive T cell will be converted into helper T cell. This helper T cell will become activated. After the activation, then this helper T cell will decide to produce those cytokines which activates those kinds of T cell or immunocells which are against a specific antigen or a specific microbes. Okay, so this is how the T cell activation is very important. But how it will be activated? It actually activated by three pathway or three signal. The first pathway we called it antigen specific stimulation pathway, or we called it signal transduction pathway. The second pathway we called it co-stimulator activation, and the third pathway we called it chemical signal or potent signal, or we called it. We will explain it later. So with the help of these three pathway, the cell become activated, and now it is ready to fight against, the infection, or it will be control the infection condition inside our body. This is a little bit a brief introduction about the T cell, that how actually it will working and perform their job inside the body. Now if we explain it with the help of diagram, normally what in disease condition, not normally, in disease condition what happen, let's suppose this is the any microbes come inside our body, let's suppose. Then what happen, this microbes will be, sometimes these microbes will infect the cell, or sometimes these microbes will be engulfed by the macrophages, or antigen presenting cell will engulf that. What happened? Let's suppose these microbes will infect the cell, or let's suppose these microbes will engulfed by the antigen presenting cell. So now this will be engulfed, as you can see. Okay, so let's suppose this is the antigen presenting cell. Antigen presenting cell. You can call that macrophages, dendritic cell, or B cell. Okay, now once they engulf this antigen or microbes, so then they will convert it into small fragment, means they digest it, as you can see. So now it's become digested. After that, this portion of the or this fragment of that antigen will be presented to their receptor, which we call that MHC2 molecule. Okay, so let's suppose this is the MHC2 molecule. MHC2 molecule. So then they will represent the antigen fragment to their receptor, which we call it MHC2 molecule. So let's suppose this is the antigen, this antigen fragment. Okay, this process, if you guys remember, we call that process antigen processing and presentation. If you guys remember, we already talked it in detail. Okay, we talked that how actually antigen is processed and presented to the rest of the cell. And we also talked that how the MHC2 molecule and MHC1 molecule are formed. It's a very huge topic. If you want to know the detail about this topic, so go back to my channel and watch this video. Now, here what happened, once the MHC2 molecule are formed and they expressing this antigen portion, so then what happened, they will secreting some kinds of cytokine which we call that interleukin 2. This interleukin 2 will just migrate or recruits the T cell. So this interleukin 2 will migrate or recruit the T cell. Okay. After that, they will secrete interleukin 2. This interleukin 2 will activate, means recruit or they migrate the T cell. So the T cell will be transferred, naive T cell. Okay, normally they will, not in disease condition, they will, recruit the naive T cell. Okay, so let's suppose this is the T cell. Naive T cell, let me write, naive T cell. Okay. This is actually the immature form of the T cell. It could be cytotoxic T lymphocyte or CD8 cell, we call it. It could be CD4 helper or helper T cell. But let's suppose in this, although we already explained cytotoxic T lymphocyte, their activation, differentiation, and in detail. If you want to watch cytotoxic T lymphocyte, it is present to my channel. You can go back and watch. Here, let's suppose we are discussing the helper T cell. Let's suppose, although I also explained the helper T cell. But what happened? This is, let's suppose, T cell receiver. They have, this is the T cell. Once they are, attracted by this interleukin 2, so then what happened? They will also, recognize this antigen portion. So for the activation, what happened? This is just, attracted. So once this T cell are reached to this macrophages, after that, what happened? This T cell will be, this T cell must be activated. Why? Because there is infection or disease inside the body. That's why this T cell must be activated. So how it will be activated? It will be activated by 3 pathways. So let me explain it. So the first pathway we call that antigen specific stimulation or we call that signal transduction pathway, or we call that signal transduction pathway. Second, we have another second pathway for the T cell activation. We call that co-stimulator signal, co-stimulation pathway. Third, we have chemical signaling, or we call that potent signaling pathway. Okay, so these are the 3 pathways. Due to this 3 pathway, the cell become activated, and after this activation, it will be differentiation into different cells. But how? Now focus here. Once this T cell are come closer, so then they will recognize this antigen through their receptor. Let's suppose they are recognizing this one receptor. Okay, so this is the receptor of the T cell. This T cell receptor is very unique in every cell. If you guys remember, we talked about the T cell receptor in detail. You know, this T cell receptor is very unique in every T cell. So this T cell receptor will recognize this antigen. So once this T cell recognizes this antigen, so then we call that antigen specific stimulation. But remember guys, the T cell receptor which recognize this antigen, which are represented by MHC2 molecule, this T cell recognize this antigen at that time when they have another co-receptor molecule which we call that CD4. Not co-receptor molecule, they have another molecule which we call it CD4. This CD4 is very important. Why? Because this CD4 is actually properly place or guide this T cell receptor that you should recognize this one receptor, or you should recognize the antigen which is represented by MHC2 molecule. Now this is the first pathway which we call it antigen specific stimulation or signal transduction pathway. Okay. Second, we have another pathway that will be activated and we call that co-stimulation. What does it mean? It means that there are some co-receptor molecule which is present with this CD4 cell. This is actually CD4 positive cell, which is present to this CD4 positive cell and to this antigen presenting cell. And the first antigen which we called it, let me remove it. So the receptor which is the co-receptor which is presented, we call that, let's suppose this is the first co-receptor. So let's suppose this is the first co-receptor which is originated from this antigen presenting cell, and we call that B7. Now this B7 receptor will be recognized by the co-receptor of the T cell, which we call that, let's suppose this is another receptor which is recognized, this B cell receptor. This receptor is called as CD28. Okay, so CD28 recognize the BC receptor of this co-receptor of this antigen presenting cell. So now the next receptor, co-receptor which is also recognized, we call that, so let's suppose this is the next co-receptor. This co-receptor is called as CD40 and it will be recognized by the CD40 ligand which is extended from this T cell. So this is actually CD40 ligand. Okay, let me once again tell you what happened after that. Co-stimulation will occur. In co-stimulation we have co-receptor molecule which is presented by this T cell as well as this antigen presenting cell. So for example, we have B7 which is recognized by the CD28. Then we have another co-receptor molecule which is the CD40 which is recognized by another co-receptor by CD40L of the T cell. So this is the co-stimulation. This co-stimulation actually tell the both of the cell that the recognition is done. Strong recognition is done. The cell is actually stabilized. Both cells are stabilized and strong recognition is done. After that, what happened? They will start secreting interleukin. Then the third pathway will activate, which we call that chemical signaling pathway. This chemical signaling pathway, during this time, different kinds of cytokines will be secreted from this antigen presenting cell as well as from this T cell. Okay. So what happened? We have for the first cytokine which we call that interleukin 12. So let's suppose these are the cytokines. And there are some receptor which we call that, there are different kinds of receptor present on the T cell. This receptor is called as interleukin 12 receiving receptor. Okay. They will actually receive the interleukin 12. So this is interleukin 12. This interleukin 12 is actually produced by this antigen presenting cell and it is received by this interleukin 12 receptor which is present in the T cell. Once they received it, so then they will give positive signal to the cell or to the nucleus of the cell. Let's suppose this is the nucleus. Inside the nucleus, RNA, DNA, means DNA is present. DNA's, central dogma process occur inside. That means DNA is converted into RNA, RNA is converted to messenger RNA, messenger RNA will produce protein. Means transcription and translation will occur. Due to transcription and translation, what happened? We have another receptor formed. Let's suppose this is another receptor. This receptor is called as interleukin 2 receiving receptor. It will be formed at that time when interleukin 12 is received. So once interleukin 2 is formed, so then the second, things that they formed, we call that interleukin 2. So then they will producing interleukin 2. So these are, let's suppose, interleukin 2. Interleukin 2, the interleukin 2 is produced by the T cell, but it will also accept by the same cell. And means this interleukin 2 will perform the function on the same cell. We call that autocrine function. So this is autocrine stimulation or function, autocrine function, okay? Or activation, whatever you can say. Once they receive it, so then this cell will be activated. Not activated after that, what happened? This cell will be secreted some other interleukins, which we call that, let's suppose this is another interleukin. We call it interleukin 1. Moreover, they will also secrete interleukin 4, interleukin 6, interleukin 7. Okay, but I'm not writing here. They will also produce another important interleukin, which we call that this is another interleukin. This is another cytokine which we call that interferon gamma. Okay. So let me once again tell you, after receiving this interleukin 2 by this interleukin 2 receptor, which we call that autocrine function, then what happened? They will secrete interleukin 1 and interferon gamma. Moreover, they are also producing some other cytokine as well, but I'm not going to write each cytokine. First of all, let's talk about this interleukin 1, what actually they will do. This interleukin 1, what actually they can do? This interleukin 1 will convert the cell into helper T cell. This interleukin 1 will convert this cell into helper T cell. So after that, then this cell will be converted to helper T cell. Interleukin 2, which is, again bind on it, they will actually make this cell, they will actually differentiate the cell, we can say like that. Differentiate the cell. What does it mean? Let me a little bit explain. Differentiate means that they will, make different copies of the T cell. From this interleukin 2, this cell will produce different copies of the cell. Like cytotoxic T cell, T lymphocyte, they will produce, first of all, they will produce effector T cell. They will make the copies of effector T cell. Let me write here. First of all, they will produce the copy of effector T cell. This effector T cell will then produce different kinds of cytokine, due to which different cells will be, formed, like cytotoxic T lymphocyte, natural killer T cell. We have, helper T cell, T regulatory cell, etc. So we can say that interleukin 2 is actually responsible for the T cell differentiation. And after that, we get the rest of the cell. So that's how the cells are differentiated. Once these things are done, then the last step of the cell activation is the interferon, means these cytokines release. After these cytokines release, so then these cytokines will, passes from this antigen presenting cell, and they will tell the antigen presenting cell that I am activated and now I am ready for my function, for my job. So then it will be, separated, and then they will perform the rest of the job. They will call other cells, other immunocells, that go and control the infection or disease inside the body. Okay. Once this cell activates, so then they will separate it, and then they will call other cells through different kinds of the cytokines. Okay, so this interferon gamma, let me once again tell you, they will tell this means that I receive all the signal which are secreted by this antigen presenting cell, and now I'm activated and now I'm ready to perform my job. This is all about the interferon gamma. So after that the cell will be activated and then they will perform their function. So this is all about the cell activation, T cell activation and their differentiation. You know, among these three pathways, the second pathway is very important. Why? Because the second pathway actually guide the cell, or they will, they will actually strongly recognize the T cell or strongly bind the T cell with this macrophages or with this antigen presenting cell. Due to this pathway, the cell will strongly recognizing the antigen with their co-receptor molecule and their receptor, their original receptor molecule. After that, the rest of the process will be going on. If this second pathway is not done, so it's very difficult that the rest of the process will be going on. Okay, so we get point that co-stimulation pathway is very important. So we also say that without this co-stimulation, the cell will never show response against the antigen. But if there is co-stimulation pathway, the cell will, perform, or they will definitely respond against the antigen or against the infection or disease. After that, the disease will be controlled. So that's all about the T cell activation and differentiation. I hope you guys get an idea about that. But if still you have any kind of question in your mind, you can write your question in the comment. I will try my best to give you answer as soon as possible. If you like this video, so make sure to hit the like button, share this video, and definitely subscribe my channel to get more interesting videos like that. Thank you so much.